Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about Introduction to Microscopy 2, Cells and Organelles.
Now, how do cells get its name? Well, the cells are in a cork. The first cells that were observed were cork. And so the person who observed those called them a cell because of the cella, or tiny rooms that the monks used to live in. And so it says like a cell. Of course, we now say a jail cell, but the cell. And then so the first cell was seen like little boxes. You could see how it looked like little jail cells here, or little rooms that monks might live in. But then they discovered that plants have cells too. They're not shaped like little boxes, but they have cells, and they have a nucleus, just like this. It's a nucleus, and they had various organelles inside. You got the Golgi Apparatus, you got the Mitochondria, rough endoplasmic reticulum. Now, if we look at a cell, here's a cell here. Cell is not as big as a universe. Here's a galaxy here, and there's a whole universe. Here's a Milky Way galaxy. You can see a cell is smaller, and you can see a cell here. A cell is not as big as a person, but it's much bigger than DNA. It's much bigger than viruses or atoms, for example. And here we can see what you can see. With electron microscopy, you can see a cell all the way to the atom. With light microscopy, you can see mitochondria, you can see eggs, and with an unaided eye, of course, we can see a person. You might be able to see frog eggs. It all depends upon which one it is. Certainly see a chicken egg. And so different types of microscopy, due to the different resolution, allow you to see different structures, and smaller structures could be seen by electron microscopy. And so the same cell that we saw before, a simple columnar epithelial cell, a similar type simple columnar cell could be seen here. This happened to be an intestine, that's in the stomach, but both of them are simple columnar cells. So cells contain organelles, like the body contains organs. So organs in a cell is like organs in the human body, organelles in cells, that is. And so if we look at the various organelles, we see the nucleus, we see nuclear pores or DNA inside there, the Golgi apparatus here in green, mitochondria, we see the plasma membrane, the nuclear membrane, we see rough endoplasmic reticulum, are some of the organelles. And so we start with the cell membrane. The cell membrane is the gatekeeper. It marks the limit of the cell, and it separates the cell from its environment. And here we can see where two cells have just divided, and they're separated by their plasma membrane. And here we can see on the intestinal absorptive cells where the plasma membrane is pushed up by these actin filaments that inside there, and the plasma membrane separates the cell from its environment. Also, we have the nucleus. Nucleus, like a computer of a cell, is the archive of the cell. It has the information. One cell has all the information to do all the functions of all the cells in the body. It's just which is expressed. And here we see the nucleus, nucleolus. These are mitochondria in through there. It's the archive of the cell. Also, we have mitochondria that produces ATP, produces energy. It's your energy production. And we see a mitochondria double membrane structure there with cristae in through there associated with making energy. And then we have the construction team. That is the rough endoplasmic reticulum. Also, the smooth endoplasmic reticulum is associated with detoxification. So you see rough ER, you have these kind of lamellar structures in through there, and then we see ribosomes on the surface to make it rough. And it's rough. Ribosomes is the one that makes protein from the messenger RNA and the transfer RNA. And then we have smooth endoplasmic reticulum in the liver to detoxify things. So there are different types of endoplasmic reticulum. Some is rough with ribosomes, some is smooth without ribosomes. And then we have the Golgi apparatus. it modifies and adds sugars, it packages things. So it's a packaging department. And here we see the Golgi apparatus. Looks like a series of pancakes that have been cut. And you'll be lots of vesicles. As these vesicles transcend one region, then another region, then another region, and then go out after the enzymes have modified the proteins that are inside there. So if we look at animal cell, here's animal cell. Here you see the nucleus, the reticuloplasmic reticulum. Centrioles, we see microtubules, actin filaments, mitochondria. We see all of these are in these animal cells, but those some or same ones are in plant cells as well. For example, each one of them has a double membrane that is a trilaminar structure. Of the membrane, you got two heads, and then the tail structure of the it's a phospholipid bilayer is what we have. In this case, it's inside there is a cell coat, and after that coat, you have a cell wall, you might say. Then there's a cell membrane, and the cell membrane is a double lipid, a layer of phospholipid. It controls the flow of water. It masks the outer limits of the cell, separates the cell from its environment. Both animal and plant cells have a double membrane in the nucleus. So the nucleus has a double membrane structure that we see around there. It houses the DNA to tell the cell what to do and how to direct its ribosomes. And here we can see how mitochondria might have been a prokaryotic cell that the DNA was enfolded from the plasma membrane. It got encircled around it in the eukaryotic cell. Prokaryotic cell is one that doesn't have a nucleus. A eukaryotic cell has a nucleus, and the enfolding of the plasma membrane might have facilitated this double membrane that we see in both plants and animals. And so to make energy, we need mitochondria. We have them both in plants and animals. They have different shapes, and we can see a double membrane. And in this case, the prokaryotic cell itself might have got involved in a eukaryotic cell and become the mitochondria. And so there's some evidence of that, because the mitochondria is the only organelle other than the nucleus that has DNA. So the cell itself might have brought in the DNA that it has, and also the DNA is circular, which is characteristic of DNA in bacteria. So the membrane-bound sacs that we have, the vacuoles that we have in mammals for intracellular digestion, release of things, generally smaller in animal cells than they are plant cells. In plant cells, the big vacuole is associated with rigidity of the plant. Here we can see small vacuoles, which are part of a secretory process of the pancreatic aster cell. Also, cytoskeletal components, you have a centriole. A centriole is associated with organized microtubules that pull chromosomes apart. Plants do not have centrioles. And here we see centriole is 9 sets of triplet microtubules, and that's what makes your centriole, which organizes things as we say. In fact, here you can see some labile microtubules readying away from the centriole. And vacuoles, lysosomes necessary for intracellular digestion of things. White blood cells, a lot of things have lysosomes that are involved in there. Here we see some lysosomes degradation. If you think about how a tadpole has to lose its tail and then gain legs, and losing of the tail part is involved in the lysosomal digestion. So a cell is composed of 3 major classes. One is membranous organelles. Those are associated with membranes, and usually these are common structures essential for the cell. It's essential for metabolic function, cell membrane, limits the cell. Rough ER makes things. Smooth ER detoxifies things. Golgi packages things. Mitochondria provides energy and lysosome for remodeling. And then there's those that are not associated with membranes, non-membranous organelles, cytoskeletal components, microtubules, microfilaments, intermediate filaments, free ribosomes. And then there's another group, inclusions. We include those, they're expendables, need not be there for the life of the cell. Nutrients could be glycogen or lipid, need not be there, but it's important for cell function, but not cell life. Pigment, melanin granules, for example, in skin. Secretory granules, zymogen granules in the pancreas are examples of these. So in summary, we want to talk about little ultrastructure things, what makes up cells, what are the organelles inside cells, and remember that when a doctor treats you, he doesn't treat you, he treats your cells. It's the cells, the unit of life. And here we see Castle Rock at Big Bend National Forest.
We want to thank the original source of diagrams, figures that may have been used during this presentation. We want to thank the original sources from which they came. That's the end of Introduction to Microscopy 2, Cells and Organelles. Thank you.